Our last group of biomolecules are the nucleic acids. So the link between the primary structure of proteins and how that's determined is what's in our DNA, which gets carried out by RNA, which are nucleic acids. So the central dogma of biology is that DNA stores information which can be copied in cell replication. The information in DNA is converted into RNA that is then read to make a protein. So we're going to talk about what are nucleic acids. So the monomers of nucleic acids are nucleotides. Nucleotides are made out of 3 parts. So in the center there is a sugar. Okay? This is a 5-carbon sugar that is attached to a nitrogenous base on one side and a phosphate group on the other. And whether this is DNA or RNA is determined by what sugar and what nitrogenous bases are present. So DNA is deoxyribonucleic acid, and RNA is ribonucleic acid. And the difference is which functional groups are present on the 2' carbon. So again, here is the 1' carbon. The 1' carbon is attached to the nitrogenous base. Okay? The 2' carbon is right here. If there is a hydroxyl group here, then that is RNA. But if the hydroxyl group has been removed, if it's been deoxified, and instead there's just a hydrogen here, that's a DNA, deoxyribonucleic acid. The 3' carbon's the same on both, 4 and 5, and are the same on both. And the 5' carbon is what's attached to the phosphate group. So one difference between DNA and RNA is the sugar. In DNA, it's deoxyribose. In RNA, it's ribose. Nucleotides are the monomers of nucleic acids. The polymers of nucleic acids are called polynucleotides. The linkage between one nucleotide and another nucleotide that leads to the formation of a polynucleotide is called a phosphodiester bond. So the phosphate group of one nucleotide is joined to the sugar of the next nucleotide. This phosphate group is on the 5' carbon. So these are pentose sugars that make up nucleotides. The 1' carbon is where the base is attached. The 2' carbon tells us whether it's DNA or RNA. 3 is here, 4 is here, 5 is here. The phosphate group is attached to the 5' carbon. And the linkage between one nucleotide and the next is the 3' carbon on one nucleotide joins to the 5' phosphate group on the next nucleotide. And the linkage between the two is called a phosphodiester bond. Now, your textbook tells you that this is formed through a dehydration reaction, but that's not exactly right, because what happens in real life is the nucleotide that joins to the growing polynucleotide is a nucleotide triphosphate. And so what leaves when the phosphodiester bond is formed is not water, but 2 phosphate groups, or a pyrophosphate. But it's okay if you put in your notes for now that it's a dehydration reaction. It can happen with a dehydration reaction if we had just a monophosphate nucleotide hooking up to the end. Alright, so one difference between DNA and RNA is which sugar is present. DNA is the deoxyribose, which has just a hydrogen on the 2' carbon. Ribose has a hydroxyl group on the 3' carbon. Another difference is which bases are present in DNA versus RNA. 3 of the bases are the same, so both DNA and RNA have cytosine, adenine, and guanine. But the thymine base is only present in DNA, whereas the uracil base is only present in RNA. So a second difference between DNA and RNA is which bases are present. DNA has a thymine base, whereas RNA has the uracil base. Another thing that I want you guys to know is what are the different bases. So some of the bases have just a single ring. The single ringed bases are called the pyrimidines. You cut the pie. The bases C, U, and T are pyrimidines. The single ringed bases are C, U, and T. Whereas the double ringed bases are the purines. So A and G are the purines, and they're present in both DNA and RNA. And a pyrimidine base always matches up with a purine base, and this is called complementary base pairing. So an A in DNA always matches up with a T in DNA. A matches with T, and a C in DNA always matches with a G in DNA. C and G go together. The only difference is during transcription, when we make RNA, an A in the DNA would match up with a U in the RNA. So the fact that a pyrimidine matches up with a purine, and A goes with T and C goes with G, or A in DNA goes with a U in RNA, is called complementary base pairing. And it's this principle that allows for the copying of DNA during DNA replication and transcription, reading the DNA to make RNA. One more difference between DNA versus RNA is the shape. DNA is a double helix. There are two polynucleotide strands that complementary base pair with each other and twist around, forming a double helix like a spiral staircase, where the sugar phosphate backbone makes up kind of the sides of the staircase, and the bases sticking into the middle make up kind of the rungs of the ladder or the steps of the stairs going up the double helix. So DNA functions as a double helix. RNA, on the other hand, functions as a single strand, a single polynucleotide. However, RNA can have different shapes even though it's a single strand. So messenger mRNA can be a single strand that's read to build a protein, but other RNAs fold back on themselves. Other RNAs have secondary structures where one base in this single strand can complementary base pair with another base in the same strand and fold back on itself. This is the case in tRNAs, which carry amino acids that are used for transcription and translation. So making sure we have all the information in our study guide for nucleic acids. The monomers of nucleic acids are called nucleotides. The polymers are called polynucleotides. And the nucleotides can be described as joined together through a dehydration reaction, although I have caveats about that. The new bond that's formed is called a phosphodiester bond. Our examples include DNA and RNA, and I want to add one more, which is ATP. ATP is adenosine triphosphate. It's an RNA nucleotide that has 3 phosphate groups attached, and its function is for energy. When that last phosphate group is released, energy is released that can drive energy necessary processes in the cell. So our functions for our nucleic acids are DNA stores information, RNA transmits that information, and nucleotide triphosphates like ATP can be used to release energy to drive cell functions. Our nucleotides are hydrophilic. They mix well with water because of the phosphate group and the nitrogen-containing bases.